Good afternoon, and thank you all so much for joining us here today. We are so excited to be representing the University System of Maryland here at the Student Spaceflight Experiment Program. And today, we'll be discussing the genetic basis of microbial motility in microgravity.
So my name is Yaniv Kazansky. I'm a junior at the University of Maryland, majoring in biochemistry and biology.
My name is Aaron Solomon. I'm also a junior at the University of Maryland studying cell biology and molecular genetics.
And our third member, Gary Sarouche, couldn't be here today, but he is also a junior like us, majoring in physiology and neurobiology.
And so for thousands of years, humans have been enchanted by space and the possibility of space exploration. And today, we are closer than ever before to being able to venture out into our solar system. But as Matt Damon discovered in The Martian, space exploration poses significant challenges to the human body. And more importantly, while our rockets may be powerful enough to carry us to Mars, space medicine still needs major advances to be able to keep astronauts healthy during long-term space travel.
Now, one of the things space medicine is going to have to address is enhanced bacterial pathogenicity in space. Now, pathogenicity simply refers to the ability of bacteria to infect us. And in order to test pathogenicity, we're going to be looking at a quality called motility, which simply refers to the ability of bacteria to move around, which we typically do using these long whip-like structures called flagella. And the ability of a bacterium to move is related to its ability to infect us. Since the easier it is for us to move, the easier it is for the bacteria to basically enter our bodies. Now, we're going to be looking at something called gene expression. So some of you may be familiar with the central dogma or central theory of biology. And what it simply states is that this molecule DNA acts as a blueprint for the cell. It's basically a code comprised of 4 letters. It is transcribed into a molecule called RNA, which looks like this. And RNA is then translated into a particular protein. And depending on what environment a bacterium is in, bacteria can turn on or express different proteins based on their situation. And past studies done on the International Space Station have actually found that bacteria can turn on virulence genes, genes responsible for infecting us in microgravity. Now, we're going to be looking specifically at genes relating to bacterial motility. And we're going to be looking at these genes because looking at the genes and the proteins that are expressed in the cell can essentially tell us what the cell is doing. Since proteins accomplish most of the functions in the cell, such as structure, catalyzing reactions, and actually moving the flagella that I mentioned earlier, looking at the proteins and the RNA in the cell responsible for motility can tell us if something different is going on there.
But Jan mentioned that there are 3 parts to this central dogma. And so the question is, why are we looking specifically at RNA at this intermediary step right here? Well, DNA doesn't actually change as gene expression changes. It's fairly constant. There are small modifications, but by and large, it doesn't reflect how we change our gene expression, while proteins are very diverse and hard to quantify. So instead, we look at RNA, this intermediary step, and we basically count how many hits we have to each gene in the bacterial genome. We do this using a complex machine called a HiSeq, an Illumina HiSeq sequencer.
Now I'm going to talk a little bit about our actual protocol with the FME. We're using a type 3 FME with 3 compartments. The first one is going to have endospores of B. subtilis, which is our bacteria. Endospores are simply dormant, dehydrated forms of bacteria that we're going to be sending up in order to avoid any effects from the launch or the reentry. The second volume is going to have Lennox broth, which is just bacteria food. And the third is going to have a reagent called RNA Protect. Now on day U-5, 5 days before undocking, we're going to open the first clamp and basically activate the bacteria by mixing them with our food, and they're going to grow for a few days. 3 days later, on day U-2, we're going to open the second clamp and unlock the third volume and mix the grown cells with RNA Protect. Now, what RNA Protect does is it kills off the cells but preserves all of the mRNA, which is what we're going to look at when we get the FME back to Earth. And we're going to have a parallel FME back on Earth undergoing the same procedures. And so once we get the bacteria on day U-10, we're going to be looking at the mRNA, and Aaron's going to tell us how we're going to be doing that.
Great. So when we sequence the bacteria, we get out a small strand like this. But we don't just get one of these. For every bacteria we have, one Bacillus subtilis has 4,000 genes, which corresponds to 4.1 million base pairs. And since we're looking at sequencing hundreds of billions to thousands of billions of bacteria, we're actually looking at data that, if each base were 1 inch long, would stretch from Earth to Mars 50 times on their own. So why bother going ourselves? So we'll have all these genes, and it'll look like a giant mass of text like this. And somehow, we have to figure out what gene each of these sequence of letters corresponds to. And so to do that, we take a reference genome, this is our original genome. We know the genome sequence of Bacillus subtilis, and the rest of them aren't really appearing here. But if you can see in red, there are some letters that correspond to this sequence right here. In blue, some letters that correspond to this sequence right here. And each of these represent the sample reads we get from our RNA sequencing. We map them to our original known sequence, which corresponds to a gene that we can analyze, which might be a motility gene or might be something to do with metabolism. This is computationally intensive, so we go to our local neighborhood supercomputer to do it for us. It's a really fun time. And once we've aligned all of that, we can compare each of the individual genes that we've sequenced. So if you look here in red, we have genes from our Earth control sample, and in blue, our space experimental sample. And we can control the expression levels of or examine the expression levels of each of these genes. And when we see that one gene is upregulated or downregulated, it gives us insight into how bacteria are modifying their transcriptome to enhance their pathogenicity. And in the end, understanding how the bacteria are modifying this transcriptome will give us insight into how we can develop new therapeutics to deal with enhanced bacterial pathogenicity. Thank you. Since antibiotic resistance is an increasing problem on Earth and will be a permanent problem for astronauts away from medical facilities in space, this is a very pertinent and important public research question, and we thank you for your time. Thank you.
And we'd like to thank Dr. Kenneth Froworth and Dr. Wade Wingler for their mentorship and help with the project, and of course to Dr. Daniel Serrano for organizing the program at UMD.
We'd also like to thank the Maryland Space Grant Consortium and the Department of Cell Biology and Molecular Genetics at the University of Maryland. In addition, we'd like to thank our community partners, the University of Maryland, Baltimore County.
We're happy to take any questions you have.
Hi, I'm Michelle from UMBC. I was wondering if you know how many genes are encoded that are predicted to control motility?
Yeah, so Bacillus subtilis, it has 4,100 genes total, and we have a suite of about 50 genes that are known to be critical in bacterial motility. Some ones are the fly genes, fly N and fly J are sort of the most common ones, but there are a suite of about 50 that are highly implicated. But since the Bacillus subtilis genome is relatively small compared to most genomes, we're sequencing every gene that exists. So researchers can also ask questions on like metabolism or whatever else they want about bacterial existence.
Okay, I have to say something. The University of Maryland system is our 4th SSEP undergraduate community. This was an SSEP experiment. And I really need to say here that, you know, what we are providing is this very simple to use flight hardware, and it's up to the students at the different grade levels to figure out how to use it. This is a university level SSEP experiment, and all of the students that are here today should recognize that this program offers continuity, a continuum of opportunity all the way through the pipeline to graduate school. Are you all figuring out where you're going to go to graduate school yet?
Yeah, we're currently applying to medical and graduate school now. So
and did I speak to both of you when we introduced the program at the University of Maryland?
I think I was at the talk. I think.
Okay, so this is really, it's so exciting for our national team to recognize that this program is truly an education pipeline initiative where folks can come in anywhere along the education pipeline all the way up to graduate school programs. So I think it's wonderful that Maryland came aboard. I don't know how many Maryland professors are here. We've got, oh, wonderful. So this is really wonderful that you all participated, and I'd like to say that this was a Mission 10 community, and you all had such a wonderful time that you just put in an implementation plan for Mission 11 to the International Space Station. So thank you so much.